We have today with us Daniel Moen. He's now assistant professor at Oklahoma State University, and the floor is yours.
All right, thanks, Ivan. Thank you all for coming today. I will point out that I was considered one of the weird biologists in the math department, so I'm not by any stretch a mathematician, but I do use a lot of models, as you'll see today. And what I'd like to talk to you about today is the general research that I do in my laboratory. I study, as Yvonne said, I study frogs. I study more specifically macroevolution, and I look at diversity in both species numbers, morphology, and I'm starting to work on biomechanics and performance. So I like to start talks by just saying, you know, why I'm a biologist. For me, I became a biologist because I used to go outside and walk around like a couple of my students here. This is in southeastern Oklahoma, where I'm a faculty member now. And we'll find things like fruits like this pawpaw and beautiful looking woodpeckers, spiders, snakes is a cottonmouth, venomous snake down there. And of course there's frogs, from big aquatic bullfrogs to small bright green tree frogs that are closely related to the tree frogs that you have here. Okay? Now we can go to another place, go to the Amazon instead. Okay? So here, if you're walking along the trail, you'll find things like beautiful flowers, and you'll find fruits, but of course different kinds of fruits. You'll find birds, different kinds of birds, spiders and caterpillars. And of course you'll also find frogs here, from these bright colored tree frogs to really cryptic. These are also a kind of tree frog, not closely related, that hang out on the leaf litter and almost impossible to see. Okay. Now, when we compare these two things, this is what's always amazed me as a biologist, and is particularly as I was a graduate student, and why I'm studying what I am today. And that is, if we compare these two places, we see pretty big differences in diversity. Okay. So if we look at species diversity, 15 frog species, that's pretty darn good. But if you go to the Amazon, we're pushing around 100 species of frogs. Okay. If you look at families, and we can think of families as kind of like evolutionary diversity, there's 54 families of frogs. 5 of them, about 10%, are found in Oklahoma, and 12 of them, about 20%, are found in the Amazon. In terms of phenotypes, we don't really know this yet. This is part of what we're working on right now. But you could probably imagine, with so few species there and so fewer families compared to the Amazon, you're going to have a lot more phenotypic diversity in the Amazon than in the Oklahoma. And of course, you know, when we're going around, we look at places and we say, you know, why is the diversity different here than it is in this other place? Oftentimes what we think about is phenotypes in terms of ecology, body form, etc. Because, you know, if you had 15 species in Oklahoma and 97 in the Amazon, and they were all cryptic species and they all looked exactly the same, you probably wouldn't be as impressed. I guess maybe academically you'd say, oh yeah, that's kind of cool, there's this cryptic speciation, but if you just go out for a hike, everything looks the same, right? So this phenotypic stuff is what particularly interests me. Okay, so one other pattern that I want to mention that'll be an important part of the talk is that when we think about groups, these different families of frogs, some of them diversify a lot and some of them diversify a little. Now, in diversification, we can talk about species numbers. In this case, we're looking at hyalid tree frogs, and here is a group that's predominantly in South America. And here's a group that's in Australia. Now, these aren't the actual species diversities, but this phylogeny represents the approximate proportions of species diversity. And the species diversity is a lot lower here in Australia, but you can see these colors. These will be important throughout the talk. They represent different styles of life in frogs. I won't go into the details of that yet because I'll have a separate slide that shows that. But you can see there's a lot more colors up here despite smaller numbers of species, and a few fewer colors here despite a lot more species. Okay, so we have one group that's got a lot of species but not as much phenotypic diversity, or I should say ecological diversity, at least in this, and a group that has fewer species and more ecological diversity. Okay, so generally in my lab what we study to try to understand why we have these differences in diversity in phenotypes and species numbers, we study a number of different things. We look at patterns of diversity in regions, communities, and clades. We also do a lot of studies, a little bit what I'll talk about here today, of ecology, morphology, and performance. That's linking the ecology that a frog has with its body form, and then its performance and functions like swimming, jumping. And then we also look at diversification and biogeography, so we try to understand these patterns of diversity in a larger geographical context. Okay? And to do these, to answer these questions, we look at, we do a number of different things. One is measure a lot of museum specimens from things that have been previously collected. We also do field work, go out and find frogs. And then with these animals, we measure things like the size of the toe pads and the webbing in the hands. We also look at the performance. Okay, so what we're looking at here, okay, so there we've got a frog. It's on a force plate, and the force plate measures the force output in 3 different dimensions, so we can get the angle of the takeoff, the amount of force it puts out. We can also calculate the velocity and the acceleration of the frog's jump. And then we take these data, and we always do phylogenetic comparative analyses, because we're almost always doing studies across many different species and using the phylogeny for both just basic doing our statistics right, but then also for modeling the evolution of these phenotypes and numbers of species. Okay, so today what I'm going to be talking about is two specific projects. One is going to be focusing on the morphology and how that relates to this microhabitat used in frogs. The second is going to be focusing on species number and how that also relates to that microhabitat. And then the last thing, I say current directions and projects in the lab about really what I'm going to be telling you about is recent field work that I've been doing. This trip here to Spain is concluding a year of field work in which I went to Madagascar, Cameroon, Mexico, and now I'm here in Spain. And so I'll be talking about my work in Madagascar to just finish the talk with some nice pictures of pretty frogs. Okay. All right. So first, talk about the phenotypes. So again, we go back to a pond, go to a pond in Oklahoma. We're going to find a number of different things in terms of what frogs are doing, where you find these adult frogs. Okay. So up here, we've got a toad that probably should look familiar because we have not too distantly related toads here in Spain. Generally living on the ground. Sometimes they burrow down. It depends on the species. We also have spadefoot toads, which look a lot like the spadefoot toads here. They're a different family, but not super distantly related. They're fossorial, so they'll burrow down and spend most of their time underground. We also have lots of frogs in the water, like this bullfrog, spends almost all of its time in the water. And then we have tree frogs. This is another hyla species like the hyla that you have here in Spain. Okay. Now if we go to Australia, what do we find? So this is kind of like the beginning of the talk, but we're focusing on frogs here. Well, if we're looking for frogs like my wife here is doing, well, actually she's looking for tadpoles. That's why her head is down. But I just took the photo to show that if we were to look in the trees, at least at night, we'd find things like big green tree frogs. We'd find frogs that are fossorial. We would find frogs that are living on the ground. And we would find frogs that are in the water. Now, again, all of these are in the same location, more or less. We search around a body of water. Okay, so the species in similar habitats, they look similar. So if we just look at the photos and compare things, we see that oftentimes, an aquatic frog, if you asked a person on the street, does this frog live in the water, they probably would be able to say yes or no. But one thing that's important when we compare these things is that we don't really, before I did this study, there wasn't a quantitative comparison of the morphology in frogs in different habitats and particularly in different places. And in particular, here if we look, we're comparing North America to Australia. Most of you wouldn't be surprised to hear that evolutionarily these are relatively distantly related, at least most of them. Okay? And so few studies have really looked across continents because generally people think the biota in a given continent or set of continents, depending on the biogeography, are independently evolving from those in a different place. Okay? So in this case, if we measure the morphology of these frogs that are in the same habitat, let's say we measure 2 frogs living in the trees and we have similar morphology, this would mean convergent evolution, and this suggests that macroevolution is a repeatable process. Now, by this, I mean that It's if you were to start let's say two lineages in two different places that had the same environment, you'd end up getting the same sort of result. Okay? So a good example of this are the cichlid fishes in the East African Rift Lakes. If you compare those of Lake Tanganyika and those to Lake Malawi, you find sets of species that are similar in both locations, and those species that look the same are similar in many aspects of their ecology. And this divergence happened in a relatively short time period. So 10 million years for a macroevolutionary study is a relatively short amount of time. But if we look more generally, this repeated evolution it's most likely if we compare lots of different groups under a number of different conditions. One, among closely related species, the cichlids, it was about 10 million years to produce about 1,000 species. So we can say they're relatively closely related. Okay? But also an important point is this also generally is found at relatively small geographical scales. So in this case, if we look at the Anolis lizards, which are famous for convergent evolution, their convergent evolution is in the Greater Antilles Islands of the Caribbean. But if you look at the rest of the distribution of Anolis all throughout Latin America, you see that you don't have similar patterns of convergent evolution and body form. So one of the main reasons why we think that this generally doesn't happen, why you don't see repeatable evolution on over large phylogenetic distances and over large geographic scales, is because the idea is that there's too many historical contingent factors that affect the evolution of a given group. So the idea is if you put two lineages in the same place, oftentimes there's certain aspects of their biology, of their genetics, of their development that don't allow them to converge in the exact same way to live in that environment. So if we could imagine an extreme case, you wouldn't expect a frog that lives in the trees to be exactly the same as a monkey that lives in the trees, because there's fundamental differences between amphibians and mammals that are not going to ever let them be the same. But in some ways you might see a few characters, but generally this really tight convergence you wouldn't expect to see at that kind of scale. Okay. Now the thing that's curious about this is that when I was a graduate student, what I did early in my days is I just took field guides of different places, Australia, Central Africa, North America, and I flipped through the pages and I noticed, these frogs that live in the same place, they kind of look the same, and it seems like it might be possible that you have this evolutionary convergence over a really large scale, over many millions of years in frogs. We're saying around 200 million years, and around the world. So from a previous study that I had done, I looked at, and actually I talked about this here 5 years ago, I did field work in China, Australia, and the Amazon, and I found relatively close phenotypic similarity between species in the same kinds of habitats. One broader question is, do you see this around the world? So if you go to more or less any location that has a decent number of frog species, do you tend to see similar sets of species that are using similar kinds of habitats and have that morphology that's similar based on that habitat? So what I did is I sampled frogs from localities around the world in museum collections and asked, okay, what do the actual microhabitats look like in these different cases? Okay. Now here is where I said I would define this microhabitat use. So when I say microhabitat, I'm saying that frogs that are terrestrial are always living on the ground, usually or often not even entering the water for reproduction, but that's not necessarily what I'm focusing on here. I'm focusing on what the adult is doing over most of its life. Aquatic and semi-aquatic frogs, we can say semi-aquatic frogs spend a lot of their time on land, but they usually escape into water. Aquatic frogs, on the other hand, are in the water more or less all the time. Arboreal frogs are those that live in the trees. Burrowing frogs down here are those that they burrow down, they spend most of their life underground. And torrent frogs are those that live in high gradient streams, fast-flowing streams. These are typically in tropical areas, and they often climb wet rocks and vegetation on the side of streams. Okay, now I chose these based on descriptions of the ecology of different species in the literature. Now I also took a phylogeny, a big phylogeny, and pruned it down to the species that I found in these 10 different communities around the world. And that produced, across these 10 communities, 167 species. And what I did was I mapped the evolution of this microhabitat on the phylogeny to understand, are the tree frogs around the world more or less closely related? Are they distantly related? How many times has this evolved? And so for this tree of 167 species, which is not that much for frogs, right? There's over 7,000 species of frogs. But this, looking across the world, will give us a sense for the frequency of transitions. And we see that these microhabitat transitions happen quite frequently. So in particular, arboreality evolved 13 independent times on this phylogeny. So that's more times than we have localities that we actually sampled. So there are multiple different independent origins of arboreality that species have in these communities. Burrowing many times, semi and fully aquatic many times. The main important thing you can get from this is that the colors are spread all across the trees, across the tree, and so we have lots of these different transitions. Okay, so this sets up the evolutionary experiment. We could say, okay, if frogs had become arboreal 13 different times, did we always get the same kind of arboreal frog? Or is it possible that those contingent factors, those historical factors, have led to, let's say, a frog whose most recent common ancestor was aquatic, and so it's its morphology is a little bit like an aquatic frog and a little bit like an arboreal frog? Or is it possible that that these two arboreal frogs are exactly the same? Okay, so to answer this question, I collected data on functional morphology from these same species. And so again, this was on museum specimens. And when I say functional morphology, I'm talking about things on the frog that it uses for using its environment. Okay, so we're talking about the size of the tips of the fingers. In arboreal frogs, that means these big toe pads, the webbing in the hands and feet, length of the arms, length of the legs, the muscle mass. So I dissected muscle mass because that's really important for jumping and swimming and just movement in general in frogs. And for the sake of analyses, this isn't super important, but I'll just mention I did principal components analysis. This allows us to take those 10 variables and look at relationships among different species in these microhabitats. We can actually look at it in a couple of different dimensions. Okay? So what we're looking at here are these principal component analyses where we take combinations of the original variables and we can look at them on single axes. And what we see, if we compare this axis to this axis, we see generally these tree frogs are kind of clustering together. The burrowing frogs are kind of clustering together. Some things like the terrestrial frogs are kind of all over the place. There's a lot more diversity there. And if we look at another axis, we see similar kinds of patterns. And so we see that generally species are clustering together if they're in the same microhabitat, but also there's a decent amount of diversity. So this sets up this question: is it possible that we have some aspects of convergent evolution that results in the clustering of species that are in the same kind of microhabitat, but also that we might have a footprint of history, meaning that some of that disparity that we see in the plot relates to the fact that some species from one family, they're kind of moving in the direction to have similar morphology as other arboreal species, but for some reason that historical aspect still leaves an effect on their morphology. Okay, so what I did is I developed a couple of different tests to look at this history versus convergence. Okay, so what I did here is I just basically asked how important is microhabitat versus the clade that a given species is in. Okay, so in this case we can look at it in terms of major clades. So in this case here on the left, this is one major group of frogs. They're the ranoid frogs. If you study frogs, that'll probably mean something. But and these are the hyloid frogs. But if you don't study frogs, the basic idea is that most of these species have diversified in the neotropics. Most of these species have diversified in the old world tropics, Africa, Southeast Asia. Okay, so we can say, is it possible that all the tree frogs in this group are similar to each other, but there's something about being in this group, some sort of morphological characteristic that's shared among all species in that group, despite what microhabitat they use, and they're a bit different than those characteristics that are shared by all species in that group? Or is it even possible that all of these different independent origins, so every time a lineage became arboreal, it is slightly different? So we can say, okay, knowing that it's arboreal doesn't help us a whole lot, okay? So I compared these different models. I used macroevolutionary models of adaptation over long periods of time. And I'll just show you real quickly what these different models are. The first model is the complete convergence model. In this case, we're saying it doesn't matter what group a frog belongs to. If it lives in the trees, it's going to be similar to another frog that lives in the trees of a different, say, a different family, different major clade. The clade matters model is the one that I showed you. It said, okay, in this example, if a frog is aquatic, and it's part of one of those major clades, it's going to look one way. If it's aquatic and it's in the other major clade, it's going to look a slightly different way. But all the aquatic frogs within each clade are going to be similar to each other. Or the all origins models. There's a number. I have the plural means that we have multiple different models here. This one says every time a frog becomes semi-aquatic or aquatic, it's different. Okay, and so I set up multiple different models, and I did these subcategories for these 3 different kinds. I couldn't do it for these 2 states because of the way that the ancestral state reconstruction works. Okay, so the punchline is that what I found was that the convergence model, the one that did not consider any historical factors at all, was the strongest by far. Okay, so in this case, what this means is that if you go to Central Africa, or you look for tree frogs here, or you look for tree frogs in Australia or in South America, you're generally going to find a frog that looks kind of like this. Okay, and this is true for terrestrial frogs, arboreal and burrowing. But one thing I didn't mention before is I had said that we have aquatic and semi-aquatic frogs. One thing that I tested in this project was whether there's actually that distinction matters morphologically. As I say, in a reference, you can find whether you can say this is an aquatic frog and this is a semi-aquatic frog, but whether or not they were morphologically different was unsure. What I found was that these actually are relatively different. This, as you might imagine, is kind of a more extreme version of this in terms of the characteristics associated with being in the water. Okay, so what I found was that convergence of body form was really strong in frogs. Now, to go back to the beginning, remember that in frogs, or in most cases, we don't see convergence at really large timescales or around the world. In this case, I was looking at communities around the world and over 150 million years. And one of the things that kind of the strange twist about this is that typically we think that you don't find convergence at such large scales because of these historical factors, but in the end, they're all frogs, and they all have this, or at least ancestrally have this kind of specific kind of development. It's possible that, and I think actually the reason why we see strong convergence in frogs around the world is because there's only so many different ways you can make a frog, which goes back all the way to frog systematists recognized this in the 50s when they were trying to understand frog phylogeny. They said, okay, well, there's not that many independent parts in frogs. In particular, frogs have a lot less bones than most tetrapods. And they're a relatively simplified version of what they could be if you compare them to fossil amphibians. So it's possible that you can change in certain ways, but you can't change to be extremely different. Okay, so one of the implications for this is the development of variation might be really important in understanding convergence. So in this case, we might say, okay, similar habitats, yeah, there's similar selective pressures, but if you only have so many different ways you can adapt to those selective pressures, you might be more likely to converge. Okay, so the next thing I wanted to look at is a paper I just finished last year, and here I'm looking at these microhabitats again, but I'm looking at them in terms of diversification. Okay, and so species diversity varies greatly among groups of organisms. Okay, so we know we have some groups that have very few living species, and of course other groups that have a huge number. But also, if we look at groups that are sisters to each other, meaning they're the exact same age, we can see these extreme differences in diversity. In this case, we've got tuataras, which is just a single or 2 species, depending on who you ask. And its sister clade is our squamates, which, I apologize, that's out of date. It's more like 10,000 species now. And so we've got huge diversity in species numbers, and these 2 groups are sisters, so they're the exact same age. So one question is, how do you get such differences, extreme differences in numbers of species over the same amount of time? Hey, now if we look at vertebrates here, we can see that the numbers of species are really different across different groups. And if we look within frogs, we see similar kind of patterns. So some families of frogs are really have high species richness, like the hyalid frogs here that have almost 1,000 species. Other groups are really species poor, like this is, I don't know if anybody recognizes this thing. Yeah, it's a totally weird group of frog that was described, well, 15 years ago now, which I think is recent. At least it seems recent to me, but I guess that means I'm getting older. But there's only 2 species in this group now, and this is a group of frogs that's a burrowing group of frogs that's found in the Western Ghats in India. Okay, but you can see if we look across species and family, at least we get a log normal distribution here in terms of the species number. Okay, so I want to know, why do we see such big differences in species number across clades? Okay, so one nice thing about analyzing diversification is that we see that diversification correlates with species diversity in frogs. That means that groups that have more diversity have higher diversification rates. So if we really want to understand what's producing these species, we need to study these diversification rates. And the nice thing about diversification rates is that we can estimate them relatively simply by just taking the log of the number of species, dividing it by the age of the group, and we get a diversification, a production of number of species over time. Now, I'll mention this is a really, the nice thing is that we only need species diversity and age to estimate the diversification rate. The not so great thing about it is that it's a really simplified way of looking at diversification, and there are some drawbacks to it as well. Okay, but the advantage here, what I'm going to run with, is that we can estimate this for even poorly sampled groups of frogs. Now, of course, if we want to understand these differences in diversification rates, the first thing that a lot of people think of is climate, right? So we know that most groups of frogs, like a lot of groups of organisms, are highest in the tropics. There's not as much diversity outside of the tropics. And so we might expect that diversification rates follow similar kinds of patterns. And those studies, in fact, have shown the same kind of thing, that if you go to warmer areas, you tend to find higher diversification rates in those families. And here's a plot from a paper that was published a few years ago. And I think by the authors, were they? No, I don't think they're associated with here. Ivan, do you know them?
Yeah,
was here.
Yeah.
Okay.
Was too.
Okay. And what they found was that as family niche width increases, diversification rate went up. Okay, so saying that groups that have broader tolerances have higher diversification rates. But what studies hadn't done up to this point was really look at this microhabitat that I've been talking about earlier. And recent studies about 1.5 years ago had been showing that this microhabitat matters in a lot of different groups of organisms. So we wanted to compare the importance of climate versus this microhabitat, these local scale factors. Okay? So what we did was we asked the relative influences of climatic niche position. That means, where is a group found? Is a group found, does it tend to be found in the tropics, the temperate zone? The rates of niche change, the rate at which the temperature characteristics and precipitation characteristics that are found in the range of a given clade, how fast do they change, and do we expect that if you adapt faster to climatic conditions, maybe you have a higher diversification rate? And then this microhabitat use, which I had mentioned earlier about whether frogs are living in the trees, on the ground, etc. So what we did is we compiled data. We took the climatic data from that previous study I mentioned by Gomez-Rodriguez. We took microhabitat data from mostly from the IUCN and AmphibiaWeb online for a large number of species. And then we got species diversity and ages of these different families. And then we calculated for these families diversification rates using these data, the proportion of species using different microhabitats using these data, and then the rates of climatic niche change, taking these data and looking at how quickly they evolve along the phylogeny. We calculated each of those for each family. Right? And so here's a graphical abstract of what we found. And I'm not going to expect you to get much out of this beyond to see that, beyond saying that here these colors represent the different microhabitats. We have a few more than before, just because of the larger scale of the study. But you can see the bars have lots of different colors. Okay, so we have lots of variation in what microhabitats are used by species in these different families. The phylogeny here has different colors. That reflects diversification rates. Red reflects higher diversification rates. Blue, cooler colors are lower diversification rates. And we can see that some families have really high rates. Some families, especially up here, have relatively low rates. And so we have variation in both of those characteristics. Okay, so we took these data and we asked what's the relative importance of these three different variables, so we can contribute how much, we can understand how much variation each of these factors are explaining in diversification rates across the families. Okay. So what we found in short with respect to microhabitat specifically, since this is the first time anyone had looked at microhabitat in frogs, was we found that all the arboreal types had an important influence on diversification rates. That is to say, arboreal, if you were arboreal, you tended to have a family that had more arboreal species tended to have higher diversification rates. Families that had more semi-arboreal species also tended to have higher diversification rates, and those that had more torrential species also tended to have higher diversification rates, whereas the other factors didn't seem to weren't particularly important. Okay, so these are all in green because they all represent some aspect of climbing. So remember, these torrential frogs, they live along high gradient streams, but they're often climbing on vegetation and wet rocks in the stream. Now, if we compare this microhabitat to these other factors, we find that overall these factors altogether explain about 2/3 of the variation in diversification rates across frog families. And in particular, if we want to compare the relative contribution of each of these factors to that 2/3, we see that about half of it is explained by microhabitat, by particularly being arboreal or not. About 1/3 of that variation is of that 2/3. Or I'll just say 1/4 of the total variation in diversification rates was explained by the rate of niche evolution. Okay, and this is niches, we're talking temperature and precipitation. And then 10% of the variation in diversification rates was explained by where you actually are found. And that's mostly, in this case, related to, it's actually a combination of precipitation and temperature. Okay, so altogether we found about 2/3 of the variation was explained by these three factors. This is important because about half of that 2/3 was explained by microhabitat, but that had never been looked at in frogs before. Okay. Of course, the thing you might be asking yourself is, okay, so I found that arboreality was important and led to higher diversification rates in frogs, but what's so big about arboreality, right? So we tend to think about diversification happening over large scales, right? And here arboreality is like where you're found in, you go to a pond, is the frog found up in the trees or is it found on the ground? And the suggestion here is that if it's found up in the trees, it's going to tend to have a higher diversification rate. So what's so important about it? Well, it's not, I'll be honest, it's not incredibly clear why arboreal frogs should have higher diversification rates than other kinds of frogs. One thing that's distinctive about this is remember that this was 3 different kinds: arboreal, semi-arboreal, and torrential. In all 3 of those cases, we're talking about adding a vertical dimension to ecological partitioning. That could be important. And most arboreal groups originated after the KT mass extinction, meaning that most of these groups are relatively young. They have high species. That's what leads to that high diversification rate. And I'm not sure what was so important about if this was actually important for becoming arboreal, or if there's some kind of reason why things that went extinct. I can't imagine that frogs were competing with dinosaurs, right? But this is one of these things that a recent paper pointed out, was that a lot of these groups are relatively originated about this time. All right. So the last thing I want to talk about is the things that we're doing in my lab right now. Both these 2 previous projects have been published. So one of the things that I talked about at the very beginning of this talk is trying to understand why We have diversity in ecology, diversity in morphology in one group versus another. We don't really have the data for the morphology yet, but we're working on that. But more generally, I showed you that there's lots of these different transitions to becoming arboreal, transitions to becoming burrowing, and a lot of studies use phylogenies to look at rates of character evolution between different states. It might be between woody and herbaceous plants. It might be between different kinds of lizards in terms of where they're living. In this case, it's in different kinds of microhabitats. And we often see that the transitions between these states are uneven, meaning going from one state to another is more common than going from that same state backwards or to a different state. And so one thing that I want to know is, why do we see these differences in the transition rates? Why do you see some changes happen more frequently than others? And I'm looking at it, of course, in terms of this microhabitat, because of what I've shown, the link to the body form, and previous studies that I've done that link it to the function in terms of how well they jump, how well they swim, etc. Okay, so we look back to these hyalid frogs again. We see that in some cases you see lots of transitions over relatively few numbers of species, and other cases you have fewer numbers of transitions over more species. So what we're doing is we're looking at this big phylogeny of frogs I mentioned earlier. We're estimating a new phylogeny, but it's probably not going to be super different than what's already known. But basically, we've got lots of microhabitat states, lots of different species. We're going to try to understand at the really big scale, how frequently do you transition from one kind to another, and what might explain that. And so we're looking at this in a number of different ways. One of the ways we're looking at the similarity in morphology of the frogs that are in the different habitats, with the idea that maybe if the morphology is really different between two different kinds of habitats, you don't have as many transitions between those over evolutionary time. Another possibility is because selection is usually favoring the ability to function well in your environment, not necessarily how you do it in terms of what body form you have. We're looking at performance. In the case of frogs, we're looking at jumping, swimming, and adhesion. These are three things that most, at least the first two, most frogs do at some point in their life. And all three are things that we can measure across different kinds of frogs. And so we're doing field work, as I mentioned. This is the tail end of a year of field work where I've done work in Madagascar, Cameroon, and here in Spain. And so I'm going to talk a little bit about my work in Cameroon to give you a sense for what we actually do when we do this field work, and to, like I said, show you a bunch of frogs. Okay, so Madagascar. Have any of you been to Madagascar? Okay, not many people have gone. It's a long way. It's really expensive to get there. It's a wonderful place, but a lot of us are familiar still about it, biogeographically, right? We know about things like lemurs and chameleons, and I mean, there's a lot of distinctive birds there, but I'll admit I don't know much about birds. There's vangas there if you like birds. Okay, so I think more or less any kind of organism, there's going to be a lot of endemicity in Madagascar. So one thing about that I want to emphasize here, or show you to give you a sense of where we did our field work, Madagascar is mostly a dry island, so about 2/3 to 3/4 of it is semi-desert or desert. So here we're looking at this land cover, and the capital, Tana, is right here. We did our field work when we get right over the mountains and you start coming down the mountains towards the coast. It gets really lush and you have really thick tropical rainforest, or at least you did a couple hundred years ago. Okay, so you can see here the elevation, and it goes as we're going down. So I went to this place called Andasi Bay. It's a nice place for doing research because one, there's a lot of reserves there, and two, there's a fair amount of ecotourism, so there's more services than your average place in Madagascar. Although this was the road right next to where we stayed, so it's still relatively undeveloped. But this is generally what you see when you go to Madagascar, okay? So here, this right here, that's native vegetation and trees. That's part of reserve. This here is the town. But if you were to go far out of town, it would still look mostly like this because Madagascar is heavily deforested. Living there is not, there's a lot of poverty in Madagascar. People use charcoal as their primary method of heating things, especially just boiling water to have safe water. And so I don't remember the exact figures, but it's certainly over 90% of native forest is completely gone. And when we drive from the capital to this place on Dasi Bay, I did not see any native vegetation over a 3-hour drive. It wasn't until we got to this area where we started to see these patches of preserve. And the preserves are actually really well kept. So well kept that if you look at Google Maps of Andasi Bay, you see the exact outlines of the reserves, which on the one hand is kind of depressing because that's the only thing that's preserved, but on the other hand, at least there's a fair amount of preservation within the reserves, which often doesn't happen in other places. Okay, so we went to go for these patches and find what kind of see what kind of frogs we could find in the patches. And it turns out that we've got a Blacktop Road that goes in between reserves. So there are actually about 7 reserves just around this community. Many of them we could walk to. And so what we did for the first couple of weeks is we just walked up and down the road when it rained, because frogs would cross between reserve patches, often going to breeding ponds. And so we hit the breeding season just right. This was in the middle of December. The rains come down. Find lots of frogs breeding. This one's guarding its eggs. And I'll show you a number of different, basically the different kinds of frogs that you find in Madagascar. Okay, so here this is Tychodina mascariniensis. So this is your typical aquatic frog, kind of like the Pelophylax here. I don't know what's a common name for Pelophylax. Okay, common frog. Okay, so. We also have tree frogs. This is a group Hyperoleida that's more diverse in mainland Africa, but there's one genus that is found in Madagascar. That's my thumb there for comparison. So these are really small frogs. These are a little bit bigger, but there's a couple different species. These tend to live up in the low brush vegetation or up in the trees. Then we also have another group of frogs. This is the most diverse one in Madagascar. So there are actually just 4 families of frogs in Madagascar. This one accounts for about 80% of the species in Madagascar, this one family. And here there's lots of different tree frogs. These are some of the most beautiful frogs I've ever seen in my life, especially these guys they're just like the greenest green you could have, and they have these beautiful red eyes with little blue on the fringe. These guys have the same kind of eyes. But then within this family, we also have other arboreal frogs. So remember I said that there are lots of origins of arboreality, independent origins in frogs. Here we've actually got this one, and these 2 are independent origins of arboreality, and that's independent from this one. So there's at least 3 independent origins within this one family in one island, which is really surprising. But then also within this family, we have a lot of terrestrial species. So these guys. And then we have semi-aquatic and torrential frogs as well. So this one up here is one that's actually pretty big, and people eat it. We didn't eat it, but we got data from it, which is better. Okay. So what we do with these frogs, so this is the performance stuff that I mentioned I haven't talked much about. We take a high-speed video camera. This is Carl. He is a PhD student, and he has a lot of experience in Madagascar. We take this camera here, and we point it towards an arena where we put the frogs, and we set the frog down. And this is an arboreal frog here, and I've got a video to give you a sense for what this looks like on the high-speed video. And watch closely. I'll get out of the way here. Watch this right here. This is the gold standard of frog jumping videos. If the frog pees, you know that it's jumping really hard, okay? Because, I mean, it sounds kind of cruel, but the truth is that, if we want to get the frog, great data, it doesn't help us if the frog just kind of does that and doesn't really motivate it. We want to get it to jump really hard. And if it jumps hard, it's because it's scared, okay? So, not too many are like that, but that's about as good as it gets. Okay, so most people think when I talk about what I do, they say, okay, you're doing frogs, and actually Rosa can attest to this. It's like, okay, it's a frog. You just put it down and Well, you try to catch frogs and they just jump away from you, right? So that's all you got to do is you just set it down, the frog jumps, and you get data and life is good. Is that true, Osa? Not always, yeah, right? Okay, so here I've got a few fun videos. It's a little bit dark here, but so sometimes the frog jumps a little bit to the side and he hits the wall. Sometimes we find, so that frog is swooping in the air. If we look at this in slow motion, you'll notice, watch that right leg. So that right leg is kicking a lot later than the left leg. What's actually happening is that when we later dissected the muscles of this frog, it was actually had a torn muscle. And so we find sometimes that these frogs are out there living, they've got some sort of injury, they seem to be fine in the wild, but we get them in the lab and we realize that they're not doing so well, at least not for what our purpose is. And then sometimes we've got a great jump and the landing is a little less than graceful, okay? So sometimes, at least when these things happen, it makes us laugh, and collecting these data is actually, it can be pretty frustrating if the frog refuses to jump as some do. Okay, so the second form of data, as I mentioned, we collect is on swimming. So here is Ruju, a student in Madagascar that worked with us. This is John Weens here. I'm my former PhD advisor and collaborator on this project. And then here is that same camera, and we just have the frog swimming down there. Same kind of setup. And then here, it's hard to see, but this is, there's a frog right there. It's on a frying pan. Most people get a kick out of seeing a frying pan with a frog on it. But here I'm using this because the nonstick surface on that pan actually has a very similar coefficient of friction to waxy rainforest leaves. Okay, so the waxy cuticle on a lot of rainforest trees. So we just take that pan, put the frog on it, and do this. And then when it falls off, we measure the angle. And then finally, of course, we kill the frogs and make museum specimens of them because we're actually looking at the musculature in their legs and how that relates to how they actually move. And so, of course, we don't release the frogs afterwards. Okay. Now, that was our. I showed you lots of frogs from our first part of our field trip in the first couple of weeks. We did do some more remote fieldwork expedition style where we took a four-wheel drive vehicle up into the mountains and went to larger patches of rainforest. Okay, so here, this is our full field team. This is Ruju, as I saw you before. There's John, me, Carl, who has a lot of experience in Madagascar, and his wife, and our local guide. And here's what it looks like if you go to actual I don't know if primary forest is necessarily what I could say here, but certainly it's native vegetation, okay, big trees. And one of the key things that we found in this place were these little frogs. You might recognize them. They're called Mantella. They're kind of like poison dart frogs. This one here is the golden Mantella. This is, you see this often in the pet trade. They're really pretty. But one of the other main things that we went is there's this plant called Pandanus that has really deep water wells, and a number of different species specialize on that plant. So one of these is FAL Suma Lateralis, I think. Lineatus. And you tend to find lots here. This plant, there were like 12 of these things. They're a really pretty gecko, day gecko. This is another thing that Madagascar is famous for. And then we find in the water wells of these, we found a couple of different specific species. This is from the Mantellidae, that one big family that I mentioned before. This is from the family Microhabitat, which is a really diverse family found around the world, and they have a decent diversity in Madagascar. So we ended up working with 9 species of Microhabitat. Some of them are funny and kind of big and fat. Some of them have this one has a really long neck. This one's a really good jumper. This frog was about that big, and it would consistently jump about 1 meter, which is pretty impressive. And the last thing that I learned from Madagascar beyond the biology is cyclones, or what cyclones are like. So when we were there, our last fieldwork, we had went to that site, and as we went there, we started getting really big rains coming in, and it turns out that the first cyclone of the year was coming in. We were actually at that field site on the 5th, so we got back, and on the 6th, it made landfall, and it hit us pretty hard. So that week was it was an interesting data collection week because we needed electricity for our camera and for our lights, but the cyclone came in, and we ended up making it work with a power generator. But we woke up the next morning, and this is what the streets were looking like. This is the river that runs through town. It's hard to see for context, but basically the river was about 6 or 7 meters taller than it normally is, and these are streets, and here I was walking in knee-deep water. But here I'll give you a little bit of a context. Before the cyclone, this was a walk to a fancy hotel at a restaurant. We'd occasionally go to this. This is what it looked like after the cyclone, completely impossible. And incidentally, we were thinking about going there to do our work because they were the only place in town that had electricity. And it's good we didn't, because we would have been trapped there for 3 or 4 days. But here, I think the most important thing was that the prospect that terrified us was, this is the bridge. This is where we stayed, and this is the exit out of the village, and we were flying out exactly 1 week from the cyclone. That's the river, and we've got about 6, 7 meters from there to the river. This is what it looked like the next morning. You can see that we've got about 20 centimeter margin. If it would have rained for another hour, we would have been stuck for a long time. Okay, so this was actually, it was a cool experience, but it was a little bit worrisome. And the funny thing is that, I guess for me, I'm originally from the middle of the United States. I'm not used to cyclones or hurricanes or any of that stuff. And these, the folks here, they were incredibly, what's the right word? Resilient? They were just like, okay, we're all right. It's going to drain in a couple days, not a big deal. And it wasn't a big deal to them. I mean, everyone was out looking and stuff, but it wasn't a big deal. So the last thing I'll say is that we did find some reptiles there. You probably want to know about other critters. I actually didn't really see many other critters. We saw lemurs that are active at night because we were out looking for frogs at night, but I didn't see much else because we were catching frogs or studying them. But we did find there's some really cool boa constrictors in Madagascar. This one's called Sanzinia is the genus, if you know snakes. And then we did find some of these charismatic reptiles of Madagascar, including your typical chameleons. And then we found this Brookesia, which are tiny chameleons. This one was about this big. Some of the most famous one is about the size of your fingernail. The adults are. Okay. We also found these leaf-tailed geckos on the plants. That was pretty cool. All right. So just to sum up, you know what? I'm just going to say, frogs are cool. They have a lot of diversity in morphology and in species, and that microhabitat has something to do with it. So we're trying to understand now the relationship between that microhabitat and the form and their body form and that function in these performance studies and how that relates to these transitions over time. Okay, and with that, I'll just briefly thank the organizations that have funded this research. A lot of support for doing the museum part of the work. I'd like to thank John Wiens. I hadn't mentioned that John was a part of those first 2 papers, the morphology and the species diversity that I talked about, and he's continuing to work with me on this larger project with the field work. And I'd like to thank Yvonne for hosting me here at Doñana, and thank you all for coming today.